histones, they are very important group of proteins, and their modifications that plays a vital role in cancer detection as well as in the tumor studies. Now, in this segment, we will discuss that how histone modifications they affect the cancer detection and the tumor detection in the human beings. Now, histone code, that is a hypothesis. There is a hypothesis which describes that the transcription of genetic information which is encoded in the DNA is in part regulated by chemical modification to histone proteins and primarily on their unstructural end. जो उनके unstructural ends हैं histone के, उनकी जो modification हो जाती है, that is known as the histone code. इसको हम कहते हैं histone code. मैं फिर repeat करता हूँ. The histone code is a hypothesis that the transcription of genetic information की transcription जो होती है encoded in the DNA, that is regulated by chemical modification. Chemical modification जो है ये उसको regulate करते हैं, उसको control करते हैं. Now, histone code will differ. Definitely ये differ होगा किस तरह? Depending upon the region of the chromatin. कि chromatin जो है, chromosome का जो chromatin है, उसके किस region के साथ है. Then the cell type, किस तरह का cell है, and the tissue type, किस तरह का tissue है, at the same time the external conditions of a cell. कि cell की जो external conditions हैं वो क्या हैं, उसमें फिर histone code will differ. Histone code क्या है? That is the gene is modified or is regulated, histone जो है, by the chemical modification. तो अब ये जो है histone code ये differ होगा altered pattern of histone modification. Histone के जो pattern हैं, जो protein के altered different pattern हैं, उनकी जब modification हो जाएगी in cancer cell, that can be sought by various ways. वो उसकी modification को हम मुख्तलिफ तरीके से ढूंढ सकते हैं. We can find out and then we can detect the tumors. पहले क्या है जी? Let's discuss acetylation of histone H4. That is the hypoacetylated in esophageal squamous cells carcinomas. उसमें क्या होगा? हमें acetylation of histone H4 cells जो है उसको ढूंढना है. Then gastric cancer, testicular cancer, acute promyelocytic leukemia जो है, उनमें acetylation of histone H4 जो है उसको हमें डिटेक्ट करना है, उसको देखना है. Then monoacetylated K16H4 is क्या है? Is commonly reduced in various types of tumors, and lower level of ACK12H4 is an indication of recurrence of prostate cancer. अगर हम इस सेगमेंट को KC1 to H4 अगर ये इंडिकेट हो जाए तो इसका मतलब ये है कि प्रोस्टेट कैंसर जो है उसकी रिकरेंस हो रही है, वो दोबारा हो सकता है, पॉसिबिलिटीज आर देयर. देन also the exposure of carcinogenic nickel. Nickel is a very carcinogenic element, and this induces a huge decrease in histone acetylation. ये acetylation को कम कर देता है, उसमें डिक्रीज कर देता है और इंडिकेट करता है कि जो है हिस्टोन उसमें क्या चेंजेस हैं. हिस्टोन H4 modification, that is also trimethylation of K20H4, which is enriched in the differential cell. जो differentiated cells होते हैं उनमें क्या है? K20H4, and it is increased with age and is commonly reduced in the cancer cell. कैंसर सेल जो हैं उनमें ये रिड्यूस हो जाता है, लेकिन एज के साथ साथ ये डिक्रीज भी हो जाता है इन द डिफरेंशियल सेल, इंक्रीज हो जाता है, बढ़ जाता है. Now recently it has been found that the low level of acetylation at the lysine 9 and 18 of histone, जो उसमें 18 और 9 के सेगमेंट है, that H3 they are associated with high recurrence of prostate cancer. जो prostate cancer है उसकी high recurrence इसमें होती है. In the other study, histone H3 acetylation has been found to be reduced in human colon, primarily tumors and in several human colon cancer cells lines. So we saw that how the different acetylation, different places of acetylation, different codons of acetylation, they affect the human cancer detection, the human tumor cell detection, and what are the different stages where they are used to detect these.